Thank you very much, Neil. May I ask all the NCL experts to come on stage, and then we have a short question and answer session. So I think Luigi, you can join us. There is you have time for a couple of questions. You have all the experts available here. Your questions could relate, let's say, to the presentations. Scott, you can join as well. Remember, you are part of EUNCL. And so you have questions about the lectures or about the procedures to apply or the type of assays and so on. So the floor is yours. I think there are some microphones available. Yes, the gentleman in the back, please.
The second last speaker, you put up that data from DLS and said you need orthogonal techniques because DLS is not reliable. Isn't the real answer that the DLS gives you misleading information? It's not democracy. You don't actually have a vote. So what you really want is not orthogonal techniques plus DLS. You just have a technique that measures it properly, which is not DLS, and then tell everybody, please don't give us DLS data because it's useless. Wouldn't that be better?
Fanny.
And the other point would be on concentration. Not everybody thinks you need concentration measurement, but some people do. That message is not very clear. I was just speaking to...
No, well, there is a reason why DLS is only on our pre-screening steps. First, we receive materials that are shipped from clients that, in most of the cases, only have performed a DLS. And we need to have a way to at least check if something is very wrong during, for example, the shipping, or if really the material it's not looking as expected. So it's just a very quick way of first seeing if something in the material that we receive is very wrong, and believe me, it happens. So, and also of checking if something goes very wrong when you apply the material in different buffers. But then after this, which is the only reason why it's still in the assay cascade, we have a full list of other techniques that we apply. So we would never use that if it was not needed just as a quick check. And for that, I mean, I don't have an answer. If a sponsor comes only with DLS and I perform another measurement, it's difficult sometimes to understand if there is something wrong or not. So this is the main reason why it is still there.
So there's a chicken and egg thing. How do you ever educate the field? So if the field was told clearly, and they're not told clearly, the limitations of that technique, use something better, then the field would move on because it spins its wheels because they don't. It's quite a simple thing to fix.
I think that my colleague, pointed out very quickly the message. Please don't use that. But as you know, it takes time.
Publicly, that's not obvious. I mean, it's obvious in this room, but if you go to a normal nanomedicine developer, they don't get that. They say, well, we have to use DLS because that's what everybody asks for.
I know, we are trying, and also our colleagues in the US are trying. We are also publishing that. There are papers out there where we are saying that, and we are planning to publish more and more papers trying to deliver the message. But as you know very well, it's not easy. We should eradicate that. I agree, but still.
A paper. Thanks from the top. Thank you.
Any other question? Yes, please.
What is the time frame for all the in vitro, in vivo, everything to finish when you say you are starting? And I would like to have a comparison. You know, I understand the first one was taking a longer time, but what is, if we are applying right now? I'm asking this question. One, it looks like it's going to take some time, and I would like to learn how long. And the second reason I'm asking this is, you're asking for a GMP-like material with all the in vivo possibly finished, this, that. You would like to have those things, but by the time we have those, we're already applying for IND. We can't wait for I don't know how long time, so I would like to learn that time period.
No, the expectation we had initially, and I think the US has encountered the same issues, we were expecting to have, let's say, high quality materials, and we just figure out that it's going to be impossible to have it. the GMP material will be only ready when you enter into clinical trial, so we'll come too late. So the expectation on the material quality is much lower than GMP. The GLP-like would fit better. Of course, endotoxin bacterial-free is the basics, and the quantity will be also a critical issue for that. In terms of timelines, as I presented, the selection process is, let's say, 3 to 4 months longest, but because there are some negotiation with regards to MTA, material transfer agreement, but most of the time it's shorter than that. A complete campaign, including the in vivo assays, should stay for 9 months, and globally the process from application step 1 until you have the report at the end of the full cascade is 1 year. Approximately, I would say, depend on how many assays you have. If we go up to in vivo, if there is no issue in between, and so on and so on. So it can be 1 year, it can be longer. We expect not to be longer, but it can be shorter as well, because sometimes you figure out you're having issues with your material, and we have to stop everything and discuss for your material to be improved, and so on and so on. Globally speaking, step 1 application up to final report is 1 year, including in vivo, but can be very short than that.
Yes, please, gentleman in the front.
So when we have a drug formulation, it should be stable at least 12 months when we want to do characterization?
This is a very good question. Stability data are key for us, and we will discuss that with the applicants anyway, because since the cascade should be run on a same batch, and as I inform you, it's about 9 months if we include in vivo, we will have to have stability data, enough stability to perform all the assays on the same batch. So a 6-month stability will be the best, even if we prolong that, which is going to be the minimum, of course, but this is also a key aspect with the endotoxin quantity and stability. These are the 3 main criteria for your material to be eligible for that.
If I may, this is one of the points where we are trying also to counseling our clients, because you have to realize that if you want to go to the clinic, your formulation has to be stable. You cannot go to the clinic with a formulation which has 2 months of stability. So if we, together with you, or you realize that your formulation is not stable in the optimal condition you have to store it, then you have to rethink a bit. So this is very important.
So your formal requirement is 6 months?
At least.
At least.
And
at least, because when you will go in clinic, as Fanny mentioned, when you will go in clinic, it has to be higher than that.
And
6 months for you?
I mean, it's a minimum. In case we're not going in vivo, it will, let's say, stay long enough for going through the whole characterization. But of course, the longer, the better.
And do you have also -80 freezers?
Yes, we do. Question behind.
Do you perform characterization on the just full formulation, or you might also do toxicity studies on specific parts like the empty carrier?
We usually perform the characterization based on the reference, which is the empty carrier. When there is an API, we have the empty carrier, the loaded carrier, and usually the API for certain assays. So we are trying to have also the precursors when you're using to set up your nanoparticles. So we're not only working on the final, let's say, end product, we're also integrating the precursors, the empty nanoparticle, the free API for the references in our assays as well.
So you will accept an empty particle for characterization?
I'm sorry, because time is limited, so we have another question. If you want, no, yes, you can, yes, afterwards. The lady in red.
Yes, because somebody brought up stability studies. Do you also support stability studies? Because a lot of people, the reason why so many people are using DLS is because this is the only thing they have. Not everybody has a cryo-EM. So the question is, do you also support stability studies to ensure that it is stable over time? And you do.
Sure.
So over the shelf life.
We are really not. It's not. It's going not to be. It's not going to be the regulatory stability studies. But and really answer that.
If you need to take it all the way to the in vivo and you start with a cascade, you need to regularly check your material so it's going to be stable through the process. So DLS is the means, as Fanny has explained, But at the same time, we have to also check on the quality, because if you're going into injecting or the means that you have designed your carrier to be used, you have to make sure that is what is you have formulated at the time that the in vivo is carried out. So yes, the stability is one point. But at the same time, we work with you with your stability, because you will have a sample in your lab that you continuously monitor with us, because as part of your batch production, you work with us and you keep exchanging information. So it's a relationship we establish, okay, from that perspective.
Not just the initial package.
Not the initial package.
So I'm sorry, time is running. And just have a couple of take-home messages. First, please contact us even if you don't feel fully ready for application, because then we can advise you and give you some tips on how to apply, so that once you will apply, it will be processed much faster. Second, the EUNCL is bad. Oh my goodness. It's back.
I would like to tell you that in Hall, Montreal, the plenary session with Professor Patrick Couvreur and Professor André Nel on cancer will start exactly at 11:30.
Okay.
So take this short break and make sure you go to the hall.
Thank you, Beat. And second, the EUNCL is performed to help you with characterizing your nanoparticles if you plan, let's say, a clinical first application to the regulatory authority for if you want to go to the market. So you have to take in, I'm referring to the discussion we had about stability and so on. You have to take to consider that all the properties of your product should take into account, let's say, that one day it will become a product on the market. So it's not, EUNCL is not, let's say, just an analytical center, distributed analytical center. We are performing and helping you, let's say, to go towards the market. So anyway, contact us at any time. Yes, everything is, of course, available on the website. We have a booth, and if you want to know more about this EU-US collaboration within EUNCL, we have a second session this afternoon where we will talk about, let's say, characterization and standards, again, because this is a EU-US collaboration. So thank you very much for joining, and see you later. Don't forget, I think there is something at 11:30. I'm not sure. So.